definitely difficult to do it backwards. Well, thank you very much, Andrea and Paul. Thank you very much. That's Paul R. as opposed to Paul W. or Paul S. Thank you very much for the opportunity to come here and be part of this. It's been a wonderful, wonderful session so far. As Andrea told you, I'm going to be talking to you about RNA flow cytometry, which is something we've been doing for the last year or so at Roswell, and something that I got involved in probably back in the maybe 2000 as something that seemed like actually probably late 90s, something that seemed really interesting and cutting edge at the time with HIV, and we probably spent about probably 6, 8, 9 months on it, only to just realize that the probes weren't any good. So it started off very frustrating, and when I finally heard that it was now available, sort of jumped into the methodology. So before I go too far, I'd like to introduce you to Roswell Park Cancer Institute, which is in beautiful Buffalo, New York. Buffalo is probably most famous for its snow, and everybody always asks me, what's the, how can you live there? What's the snow like? These people from California or Oregon just do not understand. Last year really didn't have all that much, but the year before we got something like 2 meters in 3 days. So it's sort of a plus or minus. Now I've done talks where I intermix the snow situation, but I didn't do that to you. But Roswell Park was the very first cancer center, certainly in the U.S., probably in the world. It was established in 1897 by Roswell Park. Actually, most people who come there want to know where the park is and don't realize that he was an actual individual. He was a surgeon, and at the time everybody was worried about bacterial infections and that type of thing. But in fact, Roswell said that someday cancer is going to be something that we're all going to be concerned about, and so he formed this institute called Roswell, or I think at that point it was a cancer institute. It's famous for RPMI, and for those of you who use RPMI in your cytokine assays, all the more power, all the more power to you. Actually, so it's Roswell Park Cancer Institute, but it's Roswell Park Memorial Institute Media, and the M was changed in a story that you should ask me about later on. From a memorial to cancer, because they thought cancer was a happier thought than memorial. It did a lot of, back in the 70s, it did a lot of the pioneering work with precursor BALL chemotherapy, and we're also famous or infamous, for those of you who are my age and male, maybe not so famous, for the PSA test. And that's the Institute pictured there. So we're going to talk about so to move into it, we're going to talk about the measuring mRNA expression by flow. But when you think about this in terms of a technique, really there's a lot of different ways that you can measure mRNA. There's the RT-PCR. Certainly, next-gen sequencing would be probably the most popular one right now. Northern blot, gel electrophoresis, and all the issues. And I certainly think of FISH and cytogenetics and that type of thing. And so that's probably the exception in a lot of what I'll have to say. But basically, these are all batch techniques. You're going to take the cells, you're going to harvest them, you're going to lyse them. So you're really looking at a batch technique. So they'll provide comprehensive gene analysis, but there's advantages and disadvantages to each. As I say, they're really all endpoint methods. Really, you're harvesting the cells, and a significant amount of information is lost. You can't follow the expression of genes over a period of time. They are bulk methods. You're going to take the cells and basically lyse them. So there's really nothing you can do on a correlated cell-by-cell basis. And you really can't simultaneously evaluate a lot of different parameters, multiple parameters. So the question really became, can we do this by flow? And I wouldn't be standing up here if you couldn't do that. In fact, There was some pioneering work that was done as early as 1985 by Barbara Trask, which was published in Science. And basically what Barbara did was she took, she cross-linked nuclear proteins with DMS, and then she stained them up with Hertz. Then what she took was she hybridized them with this N-acetyl-2 acetyl aminofluor, AFF, labeled mouse satellite colonies. So this is these mouse satellite sequences, which represented about 10% of the genome. So she's really looking at, and really these techniques are all early techniques, only looking at a lot of mRNA. She hybridized them, then she labeled them up with an anti-AFF antibody, and then came in with a goat anti-rabbit immunoglobulin tritzy. So really a three-step process after the cross-linking. There's the Herc staining there, and there's the mouse satellite sequences in there. So really pretty pioneering, given that, particularly back in what we're talking maybe 30 years ago. A real interesting one, or another really interesting one, was by Alan Wagner back in 1992. And what makes this interesting is the fact that what Alan did was to actually look at 3 probes simultaneously. So he took his cells and he hybridized the RNA with 5' and fluorphyll-labeled oxynucleotides. And so these were directed to histones, 18S RNA, and 28S RNA. So again, something that's going to be very prevalent in the nucleus. But what he did was he used his Psi dyes, so Psi3, I think it was Psi3, Psi3.5, and Psi5, that he attached to each of those. And so he was able to simultaneously look at those three different probes inside cells, which really, it hasn't been until just recently that we've been able to do that. I was talking about this at I think a flow course in New Mexico, and I neglected to mention this particular paper. And during the talk, Alan sent me a text to point out this reference to me. Another one, and this is actually work that came out of Roswell back in the really mid 90s. Was some work that was done by Carlton Stewart, who was a mentor of mine. And what Carl and Earl Tim did was basically to detect HER2/neu in cell lines, SKBR3 cell lines. And what he did was actually a PCR amplification. So he ran the entire, entire PCR process to measure the mRNA for HER2/neu. He used a FITC probe, and his probes were dioxygen labeled. Out of that, he got a patent, and I woke up at Carl's house maybe a year or two ago, and there in the room was a picture of the patent. I don't know if he had put the patent there so that I would see it, knowing that I was talking about RNA, or not. But this is a process for performing PCR in mammalian cells. Really, the work, they carried on this work for quite a while, but out of that, really not a whole lot happened, because the impact of going through the hybridizations, the denaturizations, and was so much. I think 25 sequences on the cells was so much that there really was nothing left of the cells by the time they got them back. Another work, which really, this is the work that I alluded to early on, which was work that was done by Bruce Patterson out of Northwestern. Basically, they looked at both DNA as well as RNA. They took the PCR, they did PCR for HIV proviral DNA, and then combined that with immunophenotyping. So this is really the first attempt to do both of those. They started off by using a biotinylated CD4. So labeling the cells up with that. Then they went through the 25 cycles of PCR with denaturization at 94, annealing at 58, and primer extension at 74. 25 cycles of that, denatured, then hybridized with their carboxyfluorescein label probe, and then they came in to label up your essentially your PCR product. And then they came in with a streptavidin PE to label up the CD4. And this is essentially what they got over here. And so after they take their single cell suspension, they do the indirect surface antibody labeling, come in with the PCR, probe it with the FISH probe, come in with the second antibody to the CD4, and then analyze by flow. And this is the type of data that they got. You've got your DNA here, CD4 on the y-axis. And what you can see are basically, at least the way I look at this, are your lymphocytes and probably your monocytes over here with the dimmer CD4. Although it could also be lymphocytes that had downregulated CD4. And you can see a population of cells, not very bright, but a population of cells that do express that had proviral DNA in the cells. The other approach, and this is the one that we played around with early on, was basically looking at a lot of oglia probes, oligonucleotide probes. And so again, your cell suspension. You can do any type of surface antibody staining that you can think of, at least in terms of when you're in terms of a direct surface staining. And then you would permeabilize and come in with a multi-probe FISH. So this is usually to fairly short sequences, or this was two fairly short sequences, several hundred of these, and they would just linearly go along the RNA and label those up. And again, sort of similar data, maybe a little bit brighter, and you can see the CD4 and then these very dim CD4 cells that are positive for the RNA. So that really was pretty much it for quite a while. And it really didn't change until just recently. So in a nutshell, attempts were made early on to measure RNA by combining PCR and flow cytometry. But the results were basically pretty disappointing due to a number of factors. First would be the higher temperature really that required to do the annealing and that type of work, which really destroyed the cells. It also caused a dim signal of noise in the individuals. For the most part, we're really only looking at one target, with the exception of the work that Wagner did, where he got into 3 different probes. And it was really limited in terms of immunophenotyping to one color. The cellular context, the shape, the size, the forward, the side scatter were really compromised because of the PCR process. And so really there were no major achievements, no major changes in the process during the 90s. And no system was really capable of doing nice correlated measurements like we're accustomed to doing by flow until 2012. And at that point, really 2 different technologies came on board. One is the Smart Flare, which is marketed by Millipore. And this is really the selling point, or the big piece about this is that you can work with live cells. And then the other one is Prime Flow or Branch DNA, which is marketed by Affymetrix, or I guess at this point by Thermo Fisher, who had really just acquired the eBioscience. So I'm going to talk about these two methodologies. Both of these platforms allow for correlated measurements of protein expression levels, both surface and intracellular, and then the transcriptional activities inside these cells. So we'll start off with the Smart Flare methodology, which is going to be able to detect mRNA in live viable cells. This was developed by Chad Merkin at Northwestern University, which is kind of interesting in the sense that Merkin and Patterson were both out of Northwestern, but I don't know of any association between the two, but really a lot of work out of Northwestern in terms of trying to detect mRNA by fluorescent techniques. What this uses is a gold nanoparticle-based system that allows for the detection of mRNA expression without harming cells. And this was published back in 2007 in the Journal of American Chemistry Society. So this is an example out of that paper. They're again using SKBR3 cells, which is the same type of model that Carl had. They're looking at survivin in this case. This is their positive cells and their control cells, and then this is their control probe, and then this is the this is looking at the survivin and the control, but in a cell line that doesn't express the survivin. So it's very clear that you can see very nice staining. Definitely some background here in the control, and there are definitely some controls that you can use to alleviate some of that. The way this method works is basically you have these smart flare probes. They consist of a gold nanoparticle here in the center. And then attached to that is a DNA sequence, which is really complementary to the mRNA that you're going to be interested in trying to detect. Attached to that is a oligonucleotide, relatively short oligonucleotide, and to it is a cyanine 3 or cyanine 5, Cy3, Cy5 label that will be released. So these are so your probe is introduced to the cells. It's endocytosed by the cells and taken up, and then inside the cells, the species of mRNA that are going to be specific for that DNA sequence, this DNA sequence, are going to have a higher affinity for the sequence than will the short oligonucleotides. So as a consequence of that, the oligonucleotides will fall off. They'll remain inside the cell, but so you'll get a structure that essentially looks like this, where the mRNA that's specific for the DNA sequence will hybridize to it, and then the free oligonucleotides will be released inside the cell. Consequently, fluorescence. And then at the end, the particle is released from the cells, exocytosed, and you basically end up with a fluorescent cell. So this requires a single overnight incubation of the cells. You can use it then to sort live cells based on the mRNA expression, and then you can use those in whatever downstream assay that you're interested in. It certainly is, particularly with the Cy3 and Cy5, it's ideal for imaging, and there's essentially no toxicity. Although Vince Shanke, who was sort of a curmudgeon at times, questioned whether or not that was true. And when I approached him, he said he had heard things about toxicities. So maybe there's something, but there's really no proof that I've ever heard of a toxic reaction. This is an example of what some data would look like. Here's some nodal mRNA, or they're looking at nodal mRNA in this manuscript, which is basically an embryonic morphogen and a member of the TGF-beta family. It's a critical factor in embryonic cell development, and in this example, they were looking at melanoma cells, where it's a regulator of growth, plasticity, and importantly, tumorigenicity. And at least the hypothesis is that it's a potential marker for metastatic potential within cells or within melanoma. And these are a number of different cell lines that have been taken up, that have been incubated with the smart flare for nodal mRNA, and you can see the staining patterns that they get. These A, B, and C are supposedly more aggressive melanomas, whereas D is a less aggressive. And so you can see the really, I think, really very, very nice staining that you get with these. So what's important about this is that it's useful for the live intact cells. Here's this is a movie that'll show you basically the uptake of a HER2/neu probe by I believe SKBR3 cells, and on the right, this is going to be your positive ones, and in about 6 hours you'll begin to see uptake. And then over here is a control where you really won't see any other. Maybe if you look up here, you'll see some non-specific. Right here in the center, or left of center, is a cell that probably has the most expression, and that one you can begin. I think right now you can begin to see some take up of the probe, and so it's basically, this is what over about a 14 hour exposure. And so you can basically follow this in time lapse, so really it's not only the uptake, but also just that you can follow this over time, which relates to being able to follow gene expression within the cells. So the process is that the probes are diluted in PBS and just added straight to your culture. If you want, you can add it to RBMI. You incubate overnight, at least 12 hours or 16 hours. Then it's quantitative or quasi-quantitative via flow cytometry or fluorescent microscopy. The signal itself is stable for up to 48 hours in the live intact cells. It's non-destructive. It can be combined with other markers, immunostaining, once the process is over, or you can use other types of reactions like the mitochondria dyes, the ROS dyes, look at ion influx or any of those things. So it's really compatible with all the more conventional types of flow cytometric applications that we would use. The process in terms of setting this up is that it's only available with a Cy3 and a Cy5, which makes it difficult. The Cy3 makes it difficult for flow unless you have a 532 or 561 because it's a relatively poor yield off of a 488. The Cy5 you can detect off of a 640 laser. Within the off-the-shelf or catalog products, there's about 1,500 probes that are available, and that grows. But you can also have ones custom made for you once you've identified provide the sequence, and that process itself takes about three to 6 weeks. It requires that the cells be actively able to endocytose, and not all cells are capable of doing that. So it comes with basically a control that I guess is really pictured here, where you have the gold nanoparticle with the sequence attached to it, and the probe directly attached to that. So if this particle is always on, it's always fluorescing. So you would incubate these particles with the cells to identify whether or not the cells are capable of endocytosing the actual nanoparticles. And that's particularly important, as one of the bigger disappointments of this is that it really doesn't work very well with hematopoietic cells. They really just aren't endocytotic. So the tumor lines, those types of cell lines, it's great for, but in terms of looking at lymphocytes, it's just not going to work. The other controls that you would consider would be just a scramble control probe, and what this is going to tell you is basically a lot about the background fluorescence that you're going to have. So in here we've got basically your nanoparticle. You have your DNA sequence, and then you have a complementary sequence to it. But this is to a completely scrambled mRNA, so that there really should be nothing expressed similar to it inside the cell. So there really would be no mRNA within the cells that would displace this oligonucleotide. And so as a consequence, this will give you a pretty good idea what the background levels are. So when I showed you up front the controls with the I believe it was with the HER2, oh no, with the survivin, in that case, that was the control that was used to define what the background would be. And then other controls that you would run, and this is really true in all the assays, is a housekeeping gene, an 18S RNA or something along that line. So these are basically probes that you would expect to find in all the cells, and you can test those to show that the signal is actually being developed by the assay. And then, of course, once you've gone through those types of controls and run those types of initial experiments, you want to in some way validate either functionally or with some other technique that what you've got here is working. So you can use a protein if one is available, so an antibody to your target or to your whatever you're trying to measure. You can confirm it by PCR on the sorted cells. You can do some sort of a functional assay, or you can do a knockdown procedure. So here's an example of different cell lines, MCF7s and HeLa. And basically, they've been targeted with an EGFR with an EGFR probe, and the MCF7s express a lower amount of EGFR than do the HeLa cells, and that's pretty clearly illustrated. I mean, it's very clearly illustrated up here with the by the smart flare. Down here, this is comparing it to an RT-PCR, and what you can see again is that you get an earlier increase with the HeLa cells than you do with the MCF7s. So there's clearly there's a correlation, or a good correlation, between what you're seeing by the smart flare versus what you're seeing by the RT-PCR. So I'm going to introduce this NANOG, which is a nuclear transcription factor. This is work that was done by Steve McEllen at the University of Alabama, and has been a collaborator on this. We've talked quite a bit and actually presented together a lot of the work. And he's done a lot of the work that I'm familiar with in terms of the smart flare. So the NANOG is a nuclear transcription factor. It's really interesting in that it really maintains, or it's involved in stemness. It's a factor that's important in maintaining pluripotency. It's involved with self-renewal of undifferentiated embryonic cells. So it works with other factors, so SOX would be one. And so it functions in concert with other factors to basically establish and maintain embryonic stem cell identity. And so Steve was interested in looking at this in some of his work. And so what we've got here is basically the Cy5 probe for the NANOG, and over here an antibody. These are unstained cells. And then this is the scramble control that I told you about, which was one that is to a irrelevant mRNA species. And then here's one to the NANOG here. And so you can see that we've got a population of stem cells. These are a cell line. Don't recall off the top of my head which cell line it is, but these are a cell line that he was using. And over here are similar controls using an antibody approach. So with unstained cells coming in with an isotype, and then here's an antibody to the NANOG itself. And so very good correlation in terms of expression. Here's then taking the cells that are positive for the NANOGs, again, the unstained, the scramble, and here's the ones that are positive, doing a sort on the ones that are positive and the ones that are negative, and then running a PCR of the sorted cells. And what you can see is that if you here's the control of the NANOG sort and the positive sort. And then here's the PCR sequence. So you can see we have a positive signal for NANOG and none in the negative cells. He also had a functional assay where he basically does this scratch test. So this is a matrigel to which he's adding his cells, and then they run a scratch down the matrigel. And in this case, these are the negatively sorted cells, and there's really no invasion of these cells into the center, which is an indication, at least to Steve, that that these cells are not embryonic in origin. Whereas in this one, these are the same scratch wound matrixal assay, but this is using the positively sorted cells. And you can see here, we've got lots of cells, but they're now beginning to invade into the center of into the center of the scratch, which is an indication that they were they had the NANOG. And then this is an example, a cool example, of looking at SI RNA, or actually a hairpin RNA knockdown in a U251s, which is the glioblastoma cell line. And these, what they were knocking out was this, basically it's a glial fibrillary acid protein, which is involved in structure and actin filaments. And what they wanted to demonstrate was that it could be knocked out. And so what you've got here is just the scramble control. Here's with the GFAP Cy3 probe. Very good staining. Virtually all the cells are stained up with the probe. And then down below we have our hairpin knockout. And you can see that they've essentially knocked out the vast majority of the signal. So it's a nice way of demonstrating that you can basically knock down or knock out the expression of a particular gene with a inhibitory RNA sequence. So basically, to summarize the smart flare approach, applications, what can I use it for? Well, you can assess RNA and protein in the same cellular preparations. It's quasi-quantify, or you can qualify relative amounts of RNA species. Cell sorting, it's ideal for cell sorting and then doing downstream applications of your product. You can do simultaneous mRNA protein visualization, as well as other probes, calcium, what have you, that we've all used. It's great for doing optimization of CRISPR clones and shRNAs and siRNAs. And of course, you can do small microRNAs as well. So bottom line, it's ideal when you want to look at live cells and you want to be able to sort on the live cells. So I guess I'd ask if there are any questions about that particular technique, or I'll move on to the branched DNA. Yeah. So the question was, endocytosis does not get into the cytoplasm. So how does the probe actually get into the cytoplasm? And I guess I don't know. I mean, I know that I know it works, but in terms of taking it up into, I don't think it's actually taken up into endocytic vesicles. I think it's actually just moves into this inside the cell. Yeah.
In terms of sensibility compared to other techniques, were you able to compare?
So in terms of branched DNA versus like
instance,
a smart flare, I don't know of anybody who's actually directly compared those, but you run into do run into the difficulty, Victor, just in terms of that you're using for flow. You're really not using optimized probes for the process. So a Cy3, Cy5 are less than ideal there. But so really, if you're thinking about it in terms of flow, I would lend more myself to the next technique, which is the branched DNA, which is going to be using elexiflores and that type of thing. Whereas if you're thinking about it in terms of sorting or imaging, then I would probably lean more towards the branched DNA or the smart flare. But I think it really comes down more to the types of questions that you want to ask.
Yes, it's about the endosomal uptake. I'm a little confused because I have a horror that these nanoparticles can just pass through passively into cells. I'm sure there's an endocytic cycle. And of course, you know that is disassembled. The endosome is disassembled and organized at cell division. And in fact, just like any father with children, you know you favour one daughter versus another very often. And as a result, you get 25% given to one daughter cell versus 75% to another. So there could be a cell cycle issue in terms of loading. And therefore, why isn't there, for example, a passive dye that you can use just to measure the extent of nanoparticle uptake as one of the denominators for actually delivery?
Yeah, so that's a good idea. Well, first off, I'm not so sure about favoring one child over the other, but that is a good idea, just in terms of being able to throw a dye in there and look at uptake as you go along. That's a good idea. Honestly, I've spent more time on the branch DNA, so I'm more familiar with that. But I don't know. Does anybody else know about the endocytic uptake? So I'll report back to you on that.
So if I understood correctly, the incubation time for this kind of probe is very long. I mean, 10 hours, 12 hours. Why mRNAs can change really more rapidly in some cases. For example, for cytokines, in 4 hours, we can have a peak for TNF, and then the transcription goes down. Don't you think then this can lead to a situation where the probe doesn't really measure the amount of mRNA at the moment when you need, but the amount at the end of the experiment when you don't have the situation you want to look at?
Yeah, so I think at some level you can measure you can measure things sequentially, but you're right. It's going to take a while for the probe to get inside the cell, and then depending on when you actually make your measurement, it's going to be a cumulative type of effect. I did show you the video. So I think really the first 6 hours or so is when the probe is being taken up, and then once you've got the probe in there, you can do a washout, right? And then you can begin to see, the actual reaction itself is going to be relatively fast, the displacement, but then you can see the accumulation and the production.
So we want to do an experiment for modulation of mRNA, how to treat cells after the probe has been taken up.
Then you can take,
Okay.
That's right. Yeah.
Okay.
Is there a temperature sensitivity for the displacement of the probe?
No, that's going to happen at 37 degrees.
37.
Yeah. Okay.
Okay.
All right, so that's really the smart flare methodology. Here's some work on the branch DNA that we've been doing, which is really just the opposite, in the sense that now you're working on fixed permeabilized types of cells, but really significant in the sense that you can look at very low copy numbers and do a lot of multiplexing with it. So what is branch DNA? Well, the commercially, it's pretty much unique to PrimeFlow, although there is another company which is marketed by Thermo Fisher, but it's also there's another company called ACD, Advanced Cell Diagnostics, I think, but ACD that does very much of the same thing with branch DNA, but their market or their focus has really been on immunofluorescence assays. So it's basically can detect up to 3 RNA transcripts, although there's a lot of interest in taking that way beyond that, and work that's going on and trying to go beyond 3 transcripts. It's basically ideal for looking at mRNAs, viral RNAs, but it has a requirement that the actual transcript be 1,000 base pairs or more. That's what they're going to do, is set up multiple probes along that base pair. It certainly can define and measure in a correlated manner individual mRNAs on a cell-by-cell basis. You can look at both gene and protein expression, and I'll show you some data with that. And it works nicely as an alternative when there's no antibody that's available. You can't find, we've all found ourselves, I'm sure, in this situation where you can't find an antibody, and this is something that you really want to measure. I'm thinking about MDSCs right now and different functional proteins that are functional molecules that I like to measure in the MDSCs, and so this is really a method that we're turning to for that. So the way it works is basically you have your mRNA sequence, and then you would come in with your target probes. Now I'm only showing 2 target probes here that are specific for a region on that target mRNA, but there's really 20 of these. So there's actually 20, or you could make 30 or 40. 20 is sort of the minimum number that you would have. So there really would be 10 of these probe sets along the mRNA. And then you come in with basically a preamplifier. A lot of specificity comes in here, and I'll show you a little bit about that in a second. And that's going to bind to the top of these Z's, or the other end of the probe sets. Then we come in with amplifiers, and these are going to. So we're basically building a Christmas tree. And these are going to have basically multiples of these that are going to bind to the amplifier. And then you come in with your floor. So by the time you're done, you've amplified the sequence about 8,000-fold, or the fluorescence from one mRNA about 8,000-fold. And so you're really able to detect, at least by microscopy, single mRNA sequences. Or species. So this is basically has a theoretical amplification of 8,000, and that's assuming that you've got 20 probe set. But if you have more, then you can amplify it even more. The procedure is pretty straightforward. You take your cell line. You can do any type of treatment that you're interested in. A lot of times we'll hit them with anti-CD3 and anti-CD28, or we might incubate them with cytokines or something like that. So you take your cells, you run your experiment, and then you need a fairly high density at about 1 million to. It's nice to have 5 million per tube. You can do any type of surface or intracellular staining that you'd like to do. The surface is straightforward. The intracellular label is actually, there's really the first part of the process within the branched DNA is a fixation and a permeabilization. So after that permeabilization, you would add your intracellular labels. And then you would sequentially incubate the cells with your target probes, your preamplifier, your amplifier, and then your labels. The downside of that is that these incubations are all pretty long. I know there's got to be optimization. I know there's optimization that could be done to these, and it probably has more to do with the buffer sets that these come with than anything in terms of just lessening the incubation time or turning up the incubator. So within each of these, there's a washing step. So by the time you're really done this process, it can be it can be a long day, a 10-hour day, or maybe within any of these, after any of these amplifications, you can stop, wash, put them in the refrigerator, and come back the next day. So at the end, of course, you're going to acquire it by immunofluorescence or on a standard flow cytometer. So here's an example of a probe set. There's a left-hand Z and a right-hand oglio or right-hand Z. If you don't have the left hand in, I'm not sure which one of these is, but it's going to be one of. No probe. If you don't add any probe at all, you're not going to get any signal. If you put in only the left hand probe, you're not going to get a signal. If you put in only the right-hand probe, you're not going to get a signal. And the reason that's important is, if you get nonspecific binding of one of these probe sets somewhere along an mRNA sequence, it really requires the complementary one in the adjacent sequence in order for that amplifier or preamplifier to be able to bind. So that's really where a lot of the sensitivity comes from, because if you don't have both of those sequences for the amplifier to bind to, then you get you will get no structure. This is, of course, the one where you have both of them, and that's illustrated up here. And then, and so that's basically the idea, and really where the sensitivity to the assay or specificity of the assay comes from. There are right now 3 different probe sets. There's one that's an Alexa 48. an Alexa 647, and an Alexa 750. Not quite sure. Actually, it was explained to me once, but still didn't think it made sense. The Type 1 is for the Alexa 647. The Type 4 is for the Alexa 488, and the Type 6 is for the Alexa 750. And really, what we're looking at here is the specificity for the mRNA is just this lower piece of the probe. The rest of this, the upper part of the Z, and then the entire amplification system is universal for the Alexa 750, or universal for the Alexa 647, or universal for the 48. So you can basically use all the reagents for this one, and only thing that you would have to be changing if you wanted to go from one mRNA species to another would be changing the actual probe set that you're working with. So this is all pretty standardized, and this is where your uniqueness comes from. I think it doesn't need to be said, but you're going to get your brightest signal off of the Alexa 647, with the Alexa 488 coming in second place, and then the Alexa 750 in third place, with really the desire to put the probe that you're most interested in is going to be the off of the Alexa 647. So we've done some work. Early on, we did some work looking at Philadelphia chromosome and different cell lines. So this is the T922 translocation associated with BCR-ABL or Philadelphia chromosome. And it's known to be highly expressed in cells that have the translocation. So BCR and ABL are going to be expressed in normal cells. We all have we all have normal copies of it. It's the translocation that causes the disease. And in cells that have the translocation, they're going to have a much higher RNA content for the BCR-ABL than in cells that don't have the translocation. So we went about asking, can we detect quantitative differences in BCR and ABL, translocation positive versus negative cell lines. We looked at the specificity and the sensitivity. And then we also did quite a bit of work combining it with protein expression. So here's the probe set. This is the BCR RNA probe, and here's the ABL probe. The BCR spans an area of 1,172, and the ABL just shy of 1,000 at 895. In healthy individuals, but you'll see that there's really no probe that is spanning the actual break point or the translocation point. So what we're going to be measuring here is both BCR and ABL. And if you look at it in healthy individuals, you're going to find messages for BCR, and you're going to find message for ABL. But in K562s, which does have the translocation, you can see that there's quite a bit, and actually you can see the co-localization on the cells as well. So here's similar data by flow. In this case, we're looking at the K562s, and we'll just do a doublet discrimination, come in with a forward and side scatter. And then what we would routinely do is use one of the housekeeping genes to eliminate anything that doesn't hybridize by setting up a positive region on that. And then here you can see in the K562s, we've got co-localization of both the BCR and the ABL. So good staining with that, and very straightforward. Here's looking at some different cell lines. We have just unstained cells, and then U937s, which it's a myeloid cell line that doesn't express or doesn't have a translocation, and then the K562s, which do. This is an ABL Alexa 546. We did some early work with one of the vendors on different types of fluorochromes. And here you can see really virtually no staining on the U937s, and dim but positive staining for ABL with the Alexa 546. Here it is with the 647, and you begin to pick up the actual staining on the U9s, but the K562s are much brighter, and that's true whether you're looking at the ABL or the BCR, which is relatively dim on the U9s, but very bright on the K562s. We extended that to some other cell lines, all the KU812s and the SUP-B15s. All of these were, all these additional ones are positive, although they don't express as high amount as the K562s, and you can see that we're basically picking that up as well, where the KU812s and the SUP-B15s are relatively dim in terms of their expression, or dimmer than the K562s, but brighter than the U937s. Here's some image stream data looking at the 18S, the BCR, and the ABLE. And what you can see is that our housekeeping gene is nicely expressed. Very good expression of the BCR, and I think you can make it out, but it's a dim shot of the ABLE. But good expression and certainly co-expression by the different cells. We looked at this by PCR. So here we're looking at the U9s, the KU812s, and the K562s. And what you can see for like BCR is that the K562s by flow, they had our highest amount of binding, as you saw. The KU812s, a lower amount, and the U9s, virtually none, for both BCR as well as for ABLE. And then here's with the PCR, and basically the results correlate very well. So we're basically seeing the same type of data off of the flow as we are seeing off of the PCR. And so the branch DNA correlates well with RT-PCR. The caveat to this, and this is really a pretty boring slide, we got our K562s and our U937s before and after. So before what and after what? Well, before we had optimized our incubator. The real caveat to this procedure is the incubator that you use. It requires that it be maintained at 40 degrees C plus or minus 1 degree. And so if your incubator is at 39 or 38, your reaction won't work. If it's a couple degrees higher than that, your reaction won't work as well. So we spent probably a good 6 months trying to get this thing optimized, and finally realized that it was all about the incubator. And that really required getting a thermometer that was a NIST standard. I was established to a NIST standard, putting it in there, and then doing quite a bit of optimization of the actual procedure. What we ended up doing was putting a metal block inside, essentially at this oven, with a little bit of water in it, so that as we open and close the door, it would really re-equilibrate really rapidly. And so the one warning that I would give to anybody as trying to set this up is that it really is worthwhile taking some time to optimize your oven or your incubator. This is some work from Emily Park at BD Biosciences, she's now at ACD. Which I think is really some beautiful work just in terms of the sensitivity of this assay. Here they're looking at BCR ABLE by flow, and then we've got some imaging results here. And this is using the Lexus 647 probe. And they took the time to basically sort each of these fractions or each of these bins, and then they did spot counts on each of those bins. So at the high end, these were K562s. In these cells at the high end, they were seeing 41 to 65 spots. At the low end, as little as 1 to 5. And what I would say is that at the low end, it's by flow, it's pretty hard to, and it's you typically would want to claim a sensitivity about 5 or 6 species. That would be as low as you would be able to detect by flow. Whereas by imaging, you can really get down to counting one species. That just has to do with dwell time, the fact that the cells aren't in the cytometer long enough, in the laser beam long enough to really get down to the 1. But good, relatively large distribution between 1 and 65 copies. But they sorted here, and then we're displaying over here at the high end, where you've got an average of 36 copies per cell, and then sort of a moderate, medium to low end down here, where they're looking at 6 to 6.2, and then scanning in here at what we call the low end, at where it's 4.5. And then if you sort on the negatives, 0.05 copies per cell. So very low copy numbers can be analyzed by RNA flow, possibly as low as 1 mRNA if you're looking at it by imaging. So here's probably the major application that's out there, at least the major commercial application, and that's looking at HIV or looking at viral loads inside cells. So here's an uninfected CD4 cells, and we're looking at HIV protein versus HIV RNA, and basically in the uninfected, nothing at all. If you infect the T cells with HIV, you get in this case about 14% of the cells that are positive for the virus. If you look at the HIV RNA, we're picking up 13% of the cells. If you sort on those cells, you can see that they're virtually all positive for the protein. So good correlation, between protein and RNA, and probably an application just in terms of being able to use the technology for measuring viral loads. Major application. Here's another example of a virus. This is hepatitis C. And really they wanted to look at the dominant relationship between these different strains of the virus. And so here's strain Y only. Here's strain, this was basically an altered, codon-altered strain, B. And then this is the two mixed together, regions were drawn. And then you can see when they co-infect, the cells are being co-infected by both of the viruses. So virtually none, or 5% of the cells are just stained with the codon altered, and maybe 1% with the only, but truthfully the vast majority. So this, in this case, you're able to say that all the cells are infected with both of the different strains. Another important application of this is basically for looking at proteins or markers that you can't detect by antibodies. So in this particular case, this is an ITP, patient with ITP, and that's a platelet disorder which can lead to excessive bleeding, and it's basically an autoimmune disease. What they did was they harvested the CD4 cells or the lymphocytes from an ITP individual with ITP, and then they stimulated them with PMA and ionomycin. And what they were looking for was the expression of IL-2 or IL-21. So in just doing standard intracellular cytokine assay, here's your unstimulated and your PMA, and you can see that for both IL-21 as well as IL-2, there's virtually no cells that are expressing either. If you hit them up with PMI ionomycin, you get good IL-2 expression. I'm sure we've all seen this, and I don't know if this was done in our PMI or not, but you can see that we get good expression. But no expression of the IL-21 is detected, so it's basically a bad antibody. If you use the branch DNA approach, again, we have virtually nothing going on in terms of in the control cells, but when we stimulate them, you can see nice IL-2 and very good IL-21. In this case, measuring with protein, in this case, measuring with the mRNA. So this is again, I think, one of the, for many of us, would be one of the applications in terms of it's an alternative when an antibody is not available. So back when this was really first introduced, 2012, 2014, the methods included a protease and methanol, which ended up with a lot of destruction of proteins and higher backgrounds. It was used to help denature and unwind and expose the mRNAs to the target probes, but it digested the proteins surrounding on the cell, surrounding it digested the proteins surrounding the nucleic acid, but it also digested a lot of the proteins on the cells. So more recently, I guess a year ago, the protease and methanol were removed, and so now it's become possible to really combine this with a lot of other markers. So here's some work looking at different lymphocyte populations. This has all gone through the branch DNA technique. Here's our gating on the, in this case it's a housekeeping gene, looking at CD8 protein, NK cells, CD56 here, and CD14, and then and then correlating CD8 protein with CD8 mRNA expression. And so you can see that we have basically a negative population here, and then clear correlation between CD8 message as well as CD8 protein. This is looking at the lymphocytes, pulling out the NK cells with the CD56. Here it is on the NK cells, and really, frankly, near perfect correlation in terms of the lower cells expressing surface CD8 correlate with the lower amount of message, and then virtually none or none on the monocytes. One of the issues, though, is that the procedure itself is tough on the cells. So these are examples of cells that have gone through either standard staining techniques, just surface staining, versus the branch DNA. So for looking at CD8 and PE, you can see we get very nice staining with the antibody alone, but when we run through the branch DNA, it's reduced. And a protein that I use a lot for just sort of testing out assays is CD45RO, and you can see that that's dramatically affected there, which is more or less what I would expect, but you can see the extent of it in that example there, and that's in PE. Another interesting piece was that it can have an effect on just the epitope. This is a CD56 stain down here, and here we're using NCAM 16.2 in FITC. Not something that I would ever really think about doing is 56 in FITC, but you can see virtually no expression here. Here it is with a PC7 using a different clone, and dim but present. But interestingly, here is the NCAM 16 clone and in the PC7, and very bright. So same clone, same clone as this one, and that's the effect on the fluorochrome. But importantly, using even the same fluorochrome, but mixing up the clone can actually have a significant impact on the stain that you see. Here's taking this to a whole different level, looking at a whole variety of different antigens, CD3, CD4, CD8, and whatever your favorite one is. And then this is the MFI of the positive cells, MFI of the negative cells, and this is going to be the stain index. And so basically here, what you can see is that the black are going to be the standard method, and the gray are going to be the branch DNA method. And so you can see that in all cases, the MFI pretty much decreases a little bit in each of these cases, for each of these antigens, whereas the negatives go up a little bit. So we're seeing a little bit of reduction in fluorescence intensity and a little bit of increase in background, and that's certainly reflected here in the stain index. Its effect on the fluorochromes is also interesting. It tends to be that the ones that are further out in the red have the least impact, whereas the ones that are further down into the blue had the most impact. What is known is that the PER-CPs do not work in this reaction, but all the other ones do. There also seems to be, and this is something that we're really looking into a lot more right this moment, interactions with the BVs, and so care has to be taken with the BVs. And it appears to be more with the particular probes that you're using that seems to be the interaction. So you just have to be careful with the BVs. So before you begin, confirm your antibodies and fluors remain resolved, and following the techniques. So here's looking at some transcription factors, EOMs and T-bet. We've taken our CD8 protein and CD56 and sort of divided up into populations that are CD8 T cells, and then different numbers of, or different NK cells with different levels of CD8 expression on them. And so here's your CD8s just correlating the protein versus the mRNA, and you've seen that before. Here's the T-bet versus the EOMs. And so you can see, and this one is gated on the CD8 cells, and so you can see we have our naives and our memory cells that are expressing both E-ohms and T-bet. Interesting here, this is looking at the R12, so these are the ones that are just negative or just positive for 56. We get this pattern here of E-ohms and T-bet, which is similar to the population here. But then when you're beginning to look at the ones that have essentially the same amount of CD8 on them, which presumably would also have CD3, you can see that we get this effector memory type of phenotype. So the big point of this, or the reason for showing this, is that it works very nicely with the intracellular proteins as well as transcription factors. And this is using the standard buffer sets that come with the assay. It's not requiring like a FOXP3 or something like that buffer set. In terms of, this is interesting. So this is, again, some data from Emily, where she's looking at co-expression of CD4 mRNA versus protein. And what we have here are the negative cells. We have the cells that are bright for CD4, and then we have another population that's intermediate for CD4 protein, but actually brighter for the mRNA. And when you think about that, you can demonstrate with the CD14 protein or CD14 antibody that these green cells are in fact the monocytes. So it's interesting that here we're seeing that the actual amount of message for the CD4s is lower than the actual amount of message in the monocytes. I'm not sure I know what the significance of that is. And it may have something to do with volume, but basically I find that really interesting. And you can basically enumerate that down below, where you've got the basically our CD14 mRNA in green, and I can't really see it from here, and maybe you probably can't see it from back there, but it turns out that with the CD4 protein, CD4 lymphocytes, that the spot count was about 5 per cell, whereas for the monocytes, it's about 9 per cell. And of course, in the ones that were negative, it's only about, it's less than 1. This is some data now looking at the correlation between interferon gamma protein and interferon mRNA over time. So doing the types of kinetic studies we talked about earlier, where the cells are being stimulated with PMA and ionomyosin. And what you can see is basically that at resting, but by about 30 minutes, we're beginning to see mRNA expression, which by 2 hours is now being expressed both by protein as well as by the mRNA, and then continuing up after 5 hours. And then here, we've got the CD4 mRNA versus interferon gamma with the CD4, and I think we've probably all seen this in the assays that we've run, where the CD4 decreases. So while we can detect lots of interferon gamma, the actual message for CD4 is decreasing as well. So it's not just that the protein is disappearing, it's actually that the whole message for the protein is disappearing in these assays. And then this is an example of taking the assay really to the extreme. Here we're looking at skin samples from a psoriasis patient and looking at FC epsilon R1 versus the control. And really what it's demonstrating is that you can do this type of assay on relatively rare cells or rare Venza cells and come up with information. With the c-Kit CD3, so basically coming in with the c-Kit cells, and you can see here the expression of the beta-2 with the FC epsilon in the cells. So to summarize, really early attempts to measure mRNA by flow. There were some very good examples, but basically it was fraught with a lot of difficulties, high backgrounds, increased autofluorescence, dim signal to noise, problems with scatter characteristics, and the really limitation in the number of parameters. But then in 2012, all this really changed with the development of the smart flare and the prime flow technologies that we discussed. These are relatively high sensitivities. With the branch DNA, we're talking about 1 to 5 copies per cell. It's basically each RNA signal is essentially a distinct spot. It's very specific with little or no background. It's certainly semi-quantitative. It can be combined with both intracellular as well as extracellular markers. It's probably very important to all of us in terms of being able to use this as a methodology when you've got a protein you want to measure but aren't able to. And then I think it has applications to viral load, stem cell epigenetics, and certainly rare cell analysis. And so with that, I'd like to acknowledge a couple of people. Katiang So, who has done the lion's share of the work at Roswell, as well as Sean Colligan, who did a lot of work looking at the protein expression. Joe Taria, who's done some of the work, as well as served as a mentor for these two. We did some work early on with Mary Beth Hanley and Emily Park out at BD's Biosciences. Emily's now gone on to advanced cell diagnostics. And then we've also done some work with Dylan Maylater, Sue Reynolds, and Castile, who's really the technical expertise behind this in terms of the prime flow assay. And then with the Smart Flare, I showed you some work that Steve McGillen had done. And so with that, I thank you and be happy to answer any additional questions that you have. Oh, and don't let me forget, you are all cordially invited to the Cyto meeting, which will be held June 10th to the 14th next year in Boston at the Heinz Convention Center. So hope you could all come out there, and remember there's opportunity for travel awards and that type of thing to the Cyto meeting. Thanks. Hey.
One question. When you show the data about CD4 in monocytes,
Yep.
I remember quite well that paper, and there were some things not completely clear. For example, the methodology was not fully displayed because of a patent present in the materials and methods, which was a little bit disappointing for reviewers. However, after that, isn't it possible that you have a problem with the spontaneous autofluorescence of monocytes? And what they saw is not really more RNA, but the result of autofluorescence.
So I know in our work that we and actually there was a slide there where we do see a slight increase in the autofluorescence of the monocytes, and we spent a lot of time trying to convince ourselves whether or not that was true or not. But honestly, it's not to the. So I do think that there is an increase in the autofluorescence, and that's. That's basically where we would put the autofluorescence is. It's not like an auto unstained tube. It's actually a tube that gets everything but the actual probes. So it has all the preamplifier, the amplifier, and the fluorochromes. So there is a slight increase, but it's really only a couple of channels. It's not at the same level as what we're seeing in that paper. So while I can't really comment on Emily. I know she was convinced of it, but the amount of autofluorescence that you're seeing in there wouldn't be, or the amount of increased fluorescence you're seeing there wouldn't necessarily be the type of autofluorescence that we would normally see.
Yeah, because there are not many explanations for that. I mean, you have more RNA, much less protein, so it's quite strange.
And I have to agree with you. I'm completely disappointed with the fact that the buffers are all proprietary, and I guess if they tell you anything, they want to kill you. But yeah. Victor.
You showed that at the moment we are actually somehow limited to only 3 fluorochromes. Do you think there are going to be some development in order to, somehow to apply the technique a bit the same way as it is applied with Luminex, so where they can do, the multiplexing of
Yeah, I think
this approach
going
of
to the
DNA
level of
branch.
a multiplex where we're talking about 30 or 40 or 50 or 100 is is probably doable in the sense that it's just building the structures, but there's been limitations that I think in terms of the floors and that type of thing. So I think what's really driven this is less the molecular biology or the DNA, making the molecular products, and just the floors that are available commercially to the different vendors to make this happen. So my understanding is, or my belief is, that there's definitely effort going on in that area to try and increase it. I know that like Fluidigm would be really interested in trying to make this happen, but when they realized that it was only a couple probes, then there was less of an interest. So there's definitely a real push to do exactly what you're trying to talk about, and it's probably just a matter of time before things are coming out like that. Yeah. Rico,
I want to go back to the slide where you show that when you treat with the buffer, you have a decrease in stain index for most of the antigens. When we use the buffer for transcription factors, for example, we see in some cases, not all the cases, but in some cases, the reduction in MFI is due to the fact that maybe the titer is not correct. The titer of the antibody is not correct. So we just titrate the antibody again, and then we can have an increased fluorescence or the same fluorescence. I don't know if it was the same in this case.
So I know that the titers for all these, we're very religious about titering antibodies and using that saturation. But in terms of the answer to your question, is those titers would have been determined based on surface staining and
then treating with the buffer.
And then treating with the buffer.
Okay.
So I it's something. It's a good point. I could go back and look at that if you've seen that. That
I
may
was
be a
just
way
curious.
But we're not using subsaturating. So for all our antibodies, we're not using subsaturating amounts. We're using saturating amounts that are
okay.
Relatively high. Yeah.
Okay, we are done. So thank you again, Paul.